sort of developmental genetics. Have we talked about enhancers? I have a feeling like the first week or two, I maybe talked a little bit about transcriptional regulation and transcription and Hox genes. And this week, we need to know a little bit about enhancers. So this is one way that cells regulate which genes get turned on and turned off. So if I remember correctly, which I'm not saying I do, we talked about the fact that in black here, there is the molecule RNA polymerase, which is the protein that reads DNA and produces an RNA transcript. That's how proteins get produced. So this is what genes do. You have to have, for every gene, there needs to be a promoter. That promoter is sort of non-specific. Every single gene that's going to get transcribed by RNA polymerase, that is read and eventually turned into a protein or stay as an RNA molecule in rare circumstances, every gene, which are contained of exons, which are the parts that encode protein, and the gray parts, introns, that don't encode protein, that comprises the gene. The promoter is basically specific DNA sequences. The promoter is defined as specific DNA sequences that bind RNA polymerase and these transcription factors that are called transcription factors initiation complex, all of these little bubbles that are immediately touching RNA polymerase, those are other proteins, but those proteins aren't a polymerase. The function of those proteins is not to make RNA. That's what polymerase does. It reads DNA, produces RNA transcripts. These initiation transcription factors, this one in particular, I've talked about before, I think, although I've taught this so many times that semesters run together, so again, step back a second. This one sort of liver-shaped blob that's touching the DNA right there near polymerase is probably TATA binding protein, or TBP. That is a transcription factor that recognizes the sequence TATA, which is why it's called TATA binding protein, because it's a protein that binds TATA. And so that sequence, TATA, is a critical part in the promoter. In other words, promoter, general. Every gene has a promoter. Every gene that gets transcribed has to have this DNA sequence in it, TATA, because that's what this protein, TATA binding protein, binds. That's the first protein that binds DNA when transcription's about to start. So, steps, in order. DNA to be transcribed. Step one, TATA binding protein binds. It's actually a little bit longer of a sequence than just TATA, but this is the essential part. TATA, there's some other nucleotides as well. They're not as easy to remember. And when TBP binds DNA, then TBP recruits, which is a fancy word for saying when that randomly diffusing protein through the cell happens to stick to TBP in the right place, recruits, binds to and holds at the promoter these other proteins. And all together, these initiation complex proteins then recruit or grab and hold at the promoter RNA polymerase. So it's a stepwise process, proteins binding proteins, and eventually all these proteins together have enough strength and specificity to grab RNA polymerase and say, here, this is the part of DNA you should transcribe, assuming that proteins have brains and can think. That's what they would be saying to each other. That is how every case of transcription starts, but I'm pretty sure we've already talked about the fact that you don't want every gene to be transcribed all the time in every cell. We've got 30,000 genes in our genome. With regard to developmental biology, some of them specify skin, some of them specify this cell should be hair, muscle, and so forth. So it would be really confusing to the cell if all the genes telling the cell what to do were always turned on at the same time. So related to transcription control then, the control doesn't come from the promoter, this region that is in front of every gene, because there's no gene by gene control of expression if you change things that are happening at the promoter. the way that this gets controlled is by controlling what happens at enhancers. Enhancers are also DNA sequences, just shown as colored bits of this chromosome, where it says enhancer sequence. The important thing here is that enhancer sequences are sequences that are specific, like TATA, that other proteins bind. There are lots of DNA binding proteins. Is that me? Hopefully not. Oh, it's okay. Whew. I'm always afraid that some like pop-up notification is going to show up as like my wife texting me or something. So. Anyway, not that it would be bad, but it might be weird. So these enhancer sequences bind transcription factors. Two different types of transcription factors. There are these transcription factors that bind enhancers. Each of those transcription factors is specific to a single enhancer sequence. So let's just make up names for these just for the sake of it. Let's say this is transcription factor A and transcription factor B. So transcription factor B, let's say, binds GTATC. And that's the only DNA sequence that protein will bind. And let's say transcription factor A only binds TTAAC. You don't have to remember that, it's just arbitrary DNA. The point is, every transcription factor has a specific DNA sequence it will recognize. This is where we get gene by gene control of gene expression, because these enhancer sequences are not found upstream of or downstream. Upstream is usually to the left of genes, downstream to the right. These enhancer sequences aren't like promoters in front of every gene. They're only upstream or in front of some genes. And, more importantly, there are way more transcription factors, not these general transcription factors that bind the promoter, but these gene-specific or gene set-specific transcription factors bind in combinations with each other. So here we have a gene that's being turned on by the presence of transcription factor A binding site and transcription factor B binding site. So this is a gene that would be turned on in cells that have protein A and protein B, or transcription factor A and transcription factor B being produced. And this is even oversimplified because there are lots more types of enhancers and promoters, transcription factors, that are specific for certain sets of genes. So hopefully, the take-home message is the more transcription factors you have available in the cell, and so the more enhancer sequences that you have, the more combinations there are of those sets of transcription factors. So you can imagine genes that are turned on by ABFH transcription factors all binding their enhancers, or genes that are turned on only when transcription factor B is present, or B and J and W. There's a multitude of different combinations of transcription factors, and that's one of the ways that cells get very specific control over gene expression. And I should have already said, but you may have already guessed just by the drawing, that the role of the transcription factor proteins is similar to the role of the general transcription factors, which is they further support the placing of RNA polymerase at the promoter. So they just add more stability and structure to bringing RNA polymerase in to start transcription. So that's enhancers. Enhancers are the DNA sequences that transcription factors bind. Any questions or concerns about that?
Far away from the start of transcription. Are they like enhancers that are farther apart, or do they have a more likelihood of sort of getting deregulated and ultimately not go through that transcription?
So this is an interesting point. I don't know the specific answer to the question of, well, I do know one answer to the question of how does the distance from the gene, the transcription, the enhancer site is, how that changes gene expression. The point, I'm glad you made this, one of the points of this slide is that it shows that one enhancer site is relatively far away down the chromosome from the gene it's regulating. That's something we're going to see in this week's paper, is that enhancers can be tens of thousands, hundreds of thousands, millions of base pairs away from the genes they regulate, which has made it really hard for geneticists to try to figure out where enhancers are. And that's why a lot of geneticists very few geneticists, I should say, have figured out where enhancers are because it's hard to figure out. The promoter is easy because it's always right next to the gene. So you always know that if there's a gene here, then the promoter is going to be just 5' of that, or just to the left of the gene. But enhancers could be anywhere, and presumably they work by the DNA because it's flexible, bending, and allowing DNA sequences that are even millions of base pairs away on the chromosome from interacting with promoters of particular genes. I know that scientists have studied the question you asked, but I don't know what the answer is. So in the paper for Wednesday, you're going to see that there's one enhancer that's in an intron of another gene, for example. So it's far away, and it's part of another gene. So I won't work through this in detail. This is the point of the, in real terms, not in terms of A, B, C, D, E proteins that I made. This is an actual example of different transcription factors that specify different tissue types, if you want to look over it at your own peril. So in other words, in pancreas, you can make two different types of cells, in this example, endocrine or exocrine, and if you're expressing the P48 transcription factor, that tells those cells to become exocrine cells, but this combination of 4 transcription factors all on in the cell at the same time lead to gene expression of downstream genes that cause those cells to become endocrine genes, and then each of those endocrine cells, each of those types then has a different combination of transcription factors that tell them what kind of endocrine cells to become, and so forth. So it's this cascade of transcription factors that allows lots of different cell types to be specified.
Relatively close to the start of the process of transcription, or during that?
But it's still an enhancer. Please do, or a slow question.
I know enhancers can be either.
Another set of terms that are going to come up in this paper are cis-regulation and trans-regulation. Did I mention that in the first week of class? Don't think so? Okay, good, I guess. So let's say that we want, for whatever reason, to have no transcription of this gene at all. So we've talked before about making mutations and trying to have gene expression not occur. At least I think we saw that in last week's paper anyway. So there are 2 ways to do this. There are actually more than 2 ways, but what sorts of things, if you were godlike and you could tinker, or a molecular geneticist, and you could tinker with any of the things that you see in this picture, what could we change to make gene expression stop? That is, no transcription. Okay, so 1, you could delete the promoter. Or what in the promoter, just to lead the thinking, it's basically the same, it's an alternative. What in the promoter is important for transcription factor binding and then transcription? There's a specific DNA sequence. Ta-da. So you could delete the whole promoter, but something that would be pretty much just as effective would be mutating the TATA site. So these are basically the same thing. You could change TATA to TAT-T. You could make a single nucleotide change that would probably abolish the ability for TBP to bind that sequence, therefore it can't bind DNA, therefore it doesn't recruit the other transcription factors, therefore they don't recruit RNA polymerase, therefore no transcription. That's just one side of the equation. That's changing a DNA sequence. Well, actually, that's changing a DNA regulatory sequence. There's something else up here that we can mess with that's not DNA. If we're godlike, or molecular geneticists. What else is there up, other than DNA, illustrated? What else is illustrated up here that's not DNA? Transcription factors. So what would you do to a transcription factor to make transcription not happen?
Not bind. To enhancers.
Okay, so you could change the structure, for example, of that protein so that it doesn't bind DNA anymore.
Or make another protein that binds to the promoter?
Or, yeah, you could come up with a repressor that represses the ability of the transcription factor to bind. A little bit more complicated, but it definitely happens in nature. That is something that occurs. Where does that protein come from that we just fiddled with? That we are going to say we're making some sort of a change to the protein so it doesn't bind the DNA sequence it recognizes, its enhancer. Where'd the protein come from in the first place? I mean, it wasn't always a protein. It had to be directed to be synthesized. So the important point is that proteins are encoded by genes also. So how would we make this protein no longer bind DNA? It would start with a mutation in the gene that encodes this protein. So I don't remember what I called that. Did I call that transcription factor B, I think? Mm-hmm. Okay, so let's say the gene for transcription factor B is right there on the chromosome where we can see it. So if we make a mutation, asterisk for sort of generic change, if we make a mutation in gene B that makes it fold differently so that it can't bind DNA, then you might turn off transcription of this gene that protein B regulates. So it all comes back to DNA, but there are differences between cis-regulation and trans-regulation, and that's where we're at now. Cis means together. If you regulate a gene in cis, that means you've mutated something that's connected to or together with the gene. So that usually means if somebody mentions cis-regulation, it means there's been a change or mutation in the DNA near the gene itself, or DNA that regulates the gene that's being discussed. So this gene, whatever gene this is that's diagrammed in this figure, where it says transcription begins, let's call it gene X. You would say that if we mutated TATA, and that turned off transcription of gene X, this mutation is in a cis-regulatory element. Cis-regulatory element. The cis-regulatory element means it's DNA that controls something, like expression of the gene. Regulatory element. The cis part means that this mutation, or the effect of the mutation, is basically attached to the gene. It's part of the DNA near the gene. That's contrasted with trans-regulation. So cis means connected, together. Trans means the opposite, apart, something like that. So in this example, a trans-regulatory change would be this protein that's been mutated. We've mutated protein B. And because protein B is free to float around in the nucleus and interact with many chromosomes and many different parts of DNA, that free ability, the fact that this protein diffuses in the nucleus and can interact with lots of different chromosomes, makes proteins usually trans-regulatory elements. Meaning, and we hadn't thought about this yet because we didn't have godlike foresight, was we made this mutation in the B gene in order to turn off expression of this gene X, say. But we had unintended consequences because if we make a mutation in this promoter, the promoter is a cis-regulatory element. It only affects expression of the gene it's near. If we mutate this protein, not only does that protein now no longer bind this enhancer that regulates gene X, what else? There are other genes that have enhancers for this protein that rely on this protein to be turned on, not just gene X, because of this combinatorial cascade of gene expression regulation by lots of transcription factors. We mutate this gene, and we misfold it, or we don't. Protein B misfolds, so it can't bind DNA. Not only does it not bind this enhancer and turn off, that is, not turn on, transcription of gene X, but it floats to other chromosomes, other DNA sites that it could recognize were it able to bind DNA. And so those genes now also can't be turned on because this protein is missing. That sort of an effect is a trans effect, that a mutation in a protein, a factor that can float around and interact with multiple genes, has potentially lots of consequences besides on just one gene. That's probably enough ranting, unless you have specific questions. That's probably enough ranting from me for now about cis and trans regulation until you read about it in the context of the paper we're going to look at. Then it might make even more or less sense. Hopefully more sense. Any questions or concerns? So just, again, like usual, as you will see, like usual, different ways to think about vocabulary for the paper. If you'd like, I'd suggest typing that URL into your device. I was just sitting up here thinking that that's not actually a link in the program I'm using to present, so I can't do anything with that. But I'll plug in my laptop temporarily. Let's do this. This is a type of analysis that's going to be in this manuscript that's probably worth seeing exactly how it works. So pardon me while I type the URL into my browser. So what this program is for looking at DNA conservation between different species. It's a visualization program. So you feed into it DNA sequences from different species or different individuals. Oh, you're kidding me. You're killing me here. And I was connected to the internet. Fine. We'll do it the old-fashioned way with a laptop. So you're going to see examples of this sort of plot. In the paper. I wanted to do it on the... Okay, now to the paper itself. So that's background, specific background for the paper. We've got arms and legs. Fish don't. Well, except they have fins. Let's see, what doesn't have arms and legs? It's a terrestrial animal. Which? Snakes. Or at least, so we thought until we read this paper and we find out that there are actually some rudimentary limbs in some snakes, but not others. So here's a phylogenetic tree that is just a cartoon I drew. The ancestors of all, let's say, terrestrial animals. So as we go from past to present through time, we see that at some point we shared a common ancestor with all snakes. And then down the lineage to the right, we became eventually humans and all other sorts of first mammals and then all other sorts of mammals. So that's the kind of scale of time we're talking about here. Long ago, there were individuals that had 4 limbs, but in snakes specifically, at some point in the common ancestor of all snakes, there's a mutation or some sort of change, or maybe a set of mutations or changes, that caused them to have stubby arms and legs, limb reduction. And we know that because, we now know that, because boas and pythons, very specific species, have reduced limbs. So the ancestor had reduced limbs. Boas and pythons, we now know, retained those reduced limbs. That is, they're in their skeletons. You just can't see them when you look at this. They're tiny. You'll see pictures of this in the paper. Boas and pythons have tiny little rudimentary limbs. They don't stick out of their bodies. You can't see them unless you take like a tomography scan of the skeleton, or if you actually kill one and take out its skeleton. And down this other lineage then, that leads to what the authors refer to as wording I don't like, advanced snakes, but that's because I'm an evolutionary biologist. We don't believe that any species is more or less advanced than any other. We're just different. The advanced snakes, the non-boas and pythons, got a second mutation, or mutations, another set of mutations or changes that caused them to totally lose all evidence that they ever had ancestors that had limbs. In other words, the advanced snakes, garter snakes, cobras, and so forth, have no evidence left, really, that they have any sort of rudimentary, tiny fragments of limbs. That's evolutionary development. That's what are the mutations that cause limb reduction or limb loss. So the authors of this paper have this question: what's the genetic basis of the fact that boas and pythons still grow little tiny limbs that you can't see, but not their close relatives, close-ish relatives, the advanced snakes? And they're going to do things looking at gene expression and transcription factor binding sites and the conservation of those binding sites between, say, boas and pythons and the advanced snakes. So looking at DNA sequences of those transcription factor binding sites and making VISTA plots to try to figure out if they can identify where near these genes that they think are important in limb growth, where those mutations might have been and were those mutations in things that might be transcription factor binding sites. I hate to do this to you, so I'm not going to spend too much time on this, but these terms do show up a little bit in this paper, so it's worth defining a little bit the terminology of the development of limbs. So we've got the stylopod, the upper arm, the zygopod, the lower arm, and then the autopod, our hands. Not every organism has a hand. Some fish have fins instead of hands. So these are sort of more generic terms that developmental biologists use to refer to parts of bodies, instead of upper arm, lower arm, and so forth, because not every organism has arms, like snakes. And they're also going to talk, you can ignore the parts where they talk about anterior, posterior, proximal, distal, and dorsal ventral patterning, but it's true that if I'm going to have a hand, one of the earliest developmental biology questions, or at least among the earliest questions, was, when my arm is growing, how does it know that the palms should face in if I put my stylopods and autopods and zugopods out like this? Why isn't one arm like that and the other arm like this? Like, why don't both of the hands pattern in the same direction? Why do they pattern in a mirror image? Why are my thumbs up and not down? Those sorts of questions. Right? That's the essence of developmental biology, is how does all of this patterning happen? What proteins are involved, what genes are involved? And that's why they care a bit about, although we don't have to so much, they care about how you define the axes of limb growth. We don't need to worry about that so much.
Dr. Ross,
Yeah.
isn't it during development, it forms like when it's still, I think, like egg or zygote, they form like the gradient to it's the proximal or anterior posterior gradient. So like there's like a gene, not a gene, but there's like some things on the pole that tell the organ or right?
Yeah, so organizing centers, yeah. So there are, in early development, there are, excellent, so that's where we're headed, good segue, that there are portions of the developing tissues that basically exude either protein signals or signals to turn on certain patterns of gene expression that correspond to patterning. So for example, this dimension of my hand from palm side to knuckle side is specified by a gradient of a protein called Wnt. It's a protein that's diffusible, and if you've got, in development, there are parts of the developing limb that make a lot of this protein, Wnt, and the cells that see the most of that turn into the dorsal side, the knuckle side of the hand, and cells down here that get less of that because this protein's sending a signal this direction from high concentration to low concentration these cells then know not to turn into knuckles in that side of my hand, but to turn into the palm. And so there's a lot of work that has gone into figuring out what are the genes and proteins involved in patterning every sort of aspect of human development. So we know, for example, that the proximal distal, that is what part of the arm or leg is supposed to be shoulder to finger, is controlled by a gradient of fibroblast growth factor, FGF. And then we know that anterior-posterior patterning, like this dimension, is patterned by a gradient of a protein called sonic hedgehog, or SHH for short. We can talk later about how genes get named. And dorsal ventral is related to this Wnt pattern. So in three-dimensional space, you've got a gradient of Wnt in one axis, you've got a gradient of FGF in another axis, and you've got a gradient of sonic hedgehog in a third axis. So that gives you that gives cells a three-dimensional coordinate depending on how much any individual cell sees FGF, sonic hedgehog, and Wnt. That gives them a three-dimensional coordinate. That cell gets a particular gene expression pattern that says, oh, you're in the middle of the hand, you should be bone, sort of thing. That's the concept. So these, because they're asking about limb development, we're going to see these sorts of protein names show up in the paper. And that's why they care, because there's a lot of work that's known about what genes are important in various aspects of development. One of the types, this is just a quick slide just for developmental biology background. There are a lot of experiments that were done early in developmental biology before you had to have institutional review board approval and IACUC protocol for doing this sort of thing to animals. They determined that there were parts of, so you can imagine maybe embryo with a tiny little outgrowth that's going to turn into an arm or a leg. It's called a limb bud. Tiny little outgrowth of tissue that's eventually going to expand by cell division and turn into an arm or a leg. Part of that is called the apical ectodermal ridge. Apical means tip, ectoderm means surface, ridge means it looks like a ridge when you look at the embryo. They determined that if you removed it, this small group of cells, if you took that out of an embryo, then they would stop growing a limb. So that means that this tissue this is a lose it experiment. You get rid of that, and you stop limb development. That means this tissue is necessary for developing a limb. Get rid of it, no limb, necessary. Move it experiment, is it sufficient to cause limb growth? Is you move it somewhere else. So they could do something like add more cells from apical ectodermal ridge, and then they find that the chicken embryo, for example, grows 2 wings on one arm. Or they can put wing, so where the wing is developing, they can put the apical ectodermal ridge from a leg bud up on the wing, and then instead of a wing, the chicken grows a leg up here on a chicken, not on me. And most cool experiment, although these are just cartoons, of course, this is an experiment that is actually done. They can show that it's actually fibroblast growth factor that is the initial signal for wing development, because they could take out the apical ectodermal ridge and stick in a little pellet of just this protein, fibroblast growth factor, FGF, and that is enough to cause wing development. So that's how, in cartoon form, scientists proved that it's FGF that's the first signal that causes the embryo to know this is where I'm supposed to develop arms and wings. That's just a picture of AER. So it is literally a group of cells on a ridge on a limb bud. And those cells give chemical and gene expression signals to other cells. For example, hey, we need to grow a limb, divide a lot. That's how I got so many cells in these parts of my body. If my chest cells had kept growing, I would be a big blob about as wide as my arms are. It'd be a round ball. But these cells knew to stop growing, these cells kept growing. These cells kept dividing and growing my limbs because of signals that came from that initial apical ectodermal ridge. As it says here, keeps the mesenchymal cells in a mitotic state. Same thing. So FGF10 early tells, so this is just again, this cartoon I drew, which is why it looks so much fancier than the last cartoon we were looking at. That FGF10 expression tells the parts of the body where to start growing limbs. But after that, that FGF10 turns on a gene called Wnt that's a signaling protein. And there's this feedback loop, which is how our limbs grow so long, is that when you turn on Wnt, Wnt turns on a fibroblast growth factor 8 expression, which stimulates more FGF10 expression in the limb bud, the growing limb. And that causes more Wnt expression. So there's this feed-forward loop where expression of one gene turns into expression of another gene, which keeps each of these genes turned on and expressing protein, and that causes limb growth. Those cells just keep dividing because they're told to divide. And at some point, of course, this cycle has to stop, or else our limbs would keep growing forever. Another term you need to know is related to the apical ectodermal ridge. It's another signaling center, which is a small group of cells that instruct other cells what to do. This is called the zone of polarizing activity. They're going to talk about this a lot in this paper, ZPA. This is their main focus for trying to figure out how snakes got shortened and reduced and eventually eliminated limbs. And here's another type of experiment that could be done. This is how this zone of polarizing activity long ago was identified. Same type of thing as the AER transplant experiments. Move it to the other side of a limb bud, and then instead of developing anterior and posterior thumb to pinky access, you get duplication of the limb. You get twice as many digits to form. But when you have two ZPAs, you get 2 feet or wings forming. This is another group of cells that's important for limb development. Okay, stepping back a minute then, how does the ZPA, zone of polarizing activity, know it's the ZPA? Back to our friend sonic hedgehog, the signaling protein. It's expressed, so here's an embryo. These are the the head of the embryo is up here, anterior. These are the 4 limb buds, and the dark spots, which all of the arrowheads are pointing to, is where the sonic hedgehog transcript, the messenger RNA that encodes the protein sonic hedgehog, is turned on in this organism. So, this is anterior, head. Sonic hedgehog is expressed in the posterior, the bottom part of the limb buds. And that's important for telling the axes of the limb bud, where it's going to be the thumbs, the pinkies, and so forth. So there's a molecular signature of the ZPA. That is, gene expression dictates where the zone of polarizing activity is, and it's sonic hedgehog expression. And so you can do the same sort of experiment with leaving the original ZPA in, but putting another sort of pellet of cells that also are secreting the same protein, sonic hedgehog. And so the limb bud doesn't know which side is up, literally. And so it creates 2 of the same side of the limb. You get a mirror image hand, like with thumbs on both sides, instead of growing a normal hand. So that's, again, the type of experiment that classical developmental biologists did to figure out what are the proteins involved in patterning. Anterior, posterior, head to tail, dorsal ventral, and so forth. Cell death is also important in limb patterning. You might imagine, for example, ducks versus chickens. Ducks have webbing between their fingers, chickens don't. So, and neither do we, normally. Not always. And the way that that difference is made is that in ducks and other web-footed organisms, platypi even, there are initially cells between the digits and they don't die. They're maintained in web-footed individuals or species, but they're specifically told to die, those cells are killed by a process called apoptosis, which is programmed cell death. And that's why most of us don't have webbing between our fingers and toes, because humans gained the ability to destroy those tissues through programmed cell death. So bone morphogenesis protein, BMP, is part of this process it's part of many processes in development, but including the loss of the tissues. And so you can turn that off by proteins that inhibit BMP, including a protein called gremlin, and that's all I'm going to say about gremlin, other than it's got a cool name. But you can imagine that genes are named after the mutations they cause, usually. So you find a mutated organism, if you can figure out what the gene is that's responsible for that mutation, then normally a biologist would name the gene after the mutation that's caused by, the gene would be named after the phenotype, the trait that's caused by the mutation. I don't know what gremlin was named after, or what organism it was in, or what it actually looked like, but we could look that up probably. But so this protein inhibits bone morphogenesis protein. So you can imagine that if BMP is important in killing the cells in between the webbing, in between the digits, however, if you had gremlin that stops BMP, this protein would help to retain webbing, because it turns off BMP. BMP kills the cells in the webbing. If you add in gremlin, then that would stop cell death, retain webbing. So you can do experiments with that too. Mentioned because they talk about gremlin in this paper. So to close, we've got a few more technique-based slides that I want to go over, techniques. One is in situ hybridization. You're going to see this a lot. You've already seen some examples of it, actually. So the purpose of an in situ hybridization experiment is you want to know in an organism, where is a gene turned on? Where is a gene currently being expressed? That's a horrible organism. It already looks like it's a mutant, but it's not. So normal gene expression patterns, like that one sonic hedgehog we just saw, there are colored areas of the embryo that show that's messenger RNA for the target gene. So in situ hybridization shows us where a particular gene is being transcribed. That does not necessarily mean that the transcript is being translated into protein. Right? We always have to know what we're measuring. Here we're measuring where is the transcript of this gene. And the way that works is pretty straightforward. We know the sequence, for example, of the RNA molecule for, let's say it's sonic hedgehog. And I'm going to make up a sequence. There are U's instead of T's in RNA, so I have to remember to write U's instead of T's when I make up this sequence. Maybe I'll just not use A's and T's, or A's and, I can use A's. AACG. Okay, so that's the single-stranded messenger RNA part of it for the protein we want to detect, where is it being turned on in an embryo. The reason, by the way, that these are really useful types of experiments is because it tells you not only where in the body genes turn on, but also when. Because you can look at an embryo of any stage of development, and then you can find out how patterns of gene expression change over time. That's also something you're going to see Wednesday. Does that look like I spelled shit? There we go. Okay. So the way the in situ hybridization works is you synthesize, create an RNA molecule that, let's say in this case, this is not exactly how they do it, but for the sake of argument, for ease of of explanation. Let's say these nucleotides that make up the artificially created RNA molecule, you actually just tell a company, here, build me this RNA molecule. They synthesize it and send it to you in a tube. It has the complementary nucleotide sequence to the RNA that you want to detect. So C's pair with G's, A's pair with U's, in this case. And then you take an embryo, you fix it, and you clear it. So basically what that means is you destroy all the cell membranes, you fix it. That is, you chemically connect all of the proteins in the whole organism to each other so that the structure of the embryo stays intact. It doesn't turn into a puddle, in other words. It still looks like an embryo. Crosslink, fix all the proteins to each other. And then you can just sort of, in generic terms, you throw this labeled probe RNA onto the embryo. This probe will where the messenger RNA for the target sequence, the one we're trying to find, where is it located, will base pair where it's complementary. In other words, the probe sequence we've designed is specific for detecting only the transcript of the gene that we know we're looking for. So you get very specific, wherever in the body that target RNA is, the probe RNA will base pair with it and bind to it. And then in this sort of oversimplified example, you can detect where that probe is in the body of the fixed embryo by, in this case, looking for fluorescence. Where does the tissue glow and where does it not? Where it does not. So in all the pictures we've seen today and we will see on Wednesday, it's not fluorescence. There's actually a colorimetric reaction you can use. The probe RNA is bound to an enzyme that converts a colorless chemical into one that produces a color, and that's why those images of embryos looked like they had black spots on the embryos. So there are a number of ways to detect the RNA molecules, in other words, but the purpose is to show you where in the body of an organism the RNA molecules for a specific gene are. That tells you where that gene is being transcribed, and presumably then where the transcripts are being translated and turned into proteins. Any questions about that? So it's a technique to know when and where a gene is turned on in development. And then also when it gets turned off in development. And then also when you compare two organisms, where a gene normally gets turned on in one organism at the same stage in development, but not present in a related species at the same stage of development, like limb growth factors. If it's present in organisms that have limbs and not present in development in organisms that don't, you're pretty sure that that gene's important for limb growth and for the change in whether you have limbs or not. So it's all about looking for correlations. Oh, look, beta-galactosidase assay. So there are two different assays that the authors are specifically going to mention. These are both these methods that I'm trying not to talk about detecting where the RNA molecules are, the non-fluorescent versions. So one is you can have a messenger RNA molecule that's connected to the enzyme beta-galactosidase, which when you throw on a colorless substrate, beta-gal can process that colorless substrate into a product that has color. So it's a chemical, the RNA is linked to an enzyme that produces a chemical reaction that changes the color of something you throw onto this embryo that turns into a dark color that you can photograph and see. So that's one of the actual ways that you can label RNAs for in situ hybridization experiments. The luciferase assay is actually the assay that you use to produce light, which is another type of experiment they're going to use, so you might as well know about it. Same idea. RNA with nucleotides linked to, in this case, another enzyme that looks like Pac-Man, if anybody knows what Pac-Man is. Right. This is a classical way you draw an enzyme, I think, in most biochemistry classes. And in this case, it chews on a chemical substrate, and when that substrate is processed, by luciferase, that enzyme reaction creates light. So you actually like photograph the light coming out of an embryo in a dark room, for example. And that amount of light gives you an indication of where the target RNA molecules are that this probe molecule is bound to. So that's what a luciferase assay is. It's measuring RNA location by production instead of by chemical reaction and pigment production. Site-directed mutagenesis, they're going to mention once. I'm not even going to write anything on this slide. That means they can create custom mutations of specific types. So not just random mutations. They know that there's a particular type of mutation they want to make, and they're going to make that mutation. I will write something. You're either glazing over because you don't know what I'm talking about or because I'm boring. Either way, I don't want that to be true. So here's an example. Let's say you want to delete those 3 nucleotides because, say, you expect that those might be involved in gene expression regulation. Maybe this is an enhancer. And so you want to test whether or not those 3 nucleotides are important in gene expression regulation. So you could do site-directed mutagenesis to create an organism that does not contain those 3 nucleotides. Not science fiction, easily doable, depending on which organism you're working on. Okay, good. Last, but most importantly, because if any of these experiments are not clearly understandable, it's going to be this one. There's one experiment they're going to do called an electrophoretic mobility shift assay, but they don't call it that, they just call it an EMSA. So here I am telling you about it. Let's say that you did have site-directed mutagenesis, and there was a version of a chromosome on top that you thought had an enhancer sequence for a specific protein, transcription factor. And then let's say you did site-directed mutagenesis, and you got rid of, by deleting site-directed mutagenesis, you had the second version of the chromosome. So you got version 1, the wild type, the original version of the chromosome, site-directed mutagenesis, you get rid of the enhancer, or the thing you think is an enhancer. You got 2 different versions of this chromosome, 1 and 2, 1 with the enhancer, 2 without. And the transcription factor should bind the enhancer. EMSA is all about asking, does a protein bind to a specific nucleic acid sequence? That is, does this specific transcription factor we're working on bind this specific stretch of DNA? Sequence E, the enhancer sequence. There are a number of ways to do this sort of experiment. This is maybe the fastest or easiest. So electrophoresis is the process of separating, let's see, if we have protein samples, if we have an acrylamide gel, which is basically like a big sheet of Jell-O with holes in the top. If we load protein samples in the top of this gel and apply an electric current, which everybody remembers from physics classes, I, current, or E, voltage, because of course you wouldn't use V for voltage or C for current. You place an electric field across this gel, and different components will move through the gel depending on the type of gel and that you're running. So usually, molecular biologists think about, well,
We use 8% gels.
so you do polyacrylamide gels,
Yeah.
and that separates what? Based on what?
Protein bonds based on their size.
Okay. So you can separate proteins based on their size. So when you apply a current or an electric field across this gel, the proteins all start at the top of this piece of Jell-O, they're moving through the field, what winds up at the bottom?
The lightest.
The largest or the
smallest.
yeah, the smallest. So the smaller the protein, the more easily it can move in between the holes in the jello. So it can go farther, faster than large proteins, which are harder for electricity to pull through because proteins have electrical charges on their atoms. So they are electric, yeah. You can use electricity to pull proteins. That's the important fundamental part. So you separate something based on size. That's one approach, or one outcome of acrylamide gel electrophoresis. There are other ways to separate proteins, like based on their pH, but let's focus on protein size. So small things go to the bottom of the gel, large things are harder to pull through the gel with electricity. So if you have a gel, and you have your transcription factor plus, let's see, DNA with the enhancer, or DNA without the enhancer, and you load all of that actually in two separate wells. You got DNA transcription factor plus the DNA with the enhancer, and transcription factor plus the DNA that doesn't have the presumed enhancer sequence, and you run those separately on this gel. The one thing you have to know is you have to be able to see where the protein and the DNA is to know if they're binding each other. So this is how this process works. The DNA samples, both types, with the enhancer and without, are, say, radioactively labeled, or they're fluorescently labeled, something that lets you see where the DNA molecules are by exposing them to radiograph paper, for example, photos. If you add in, let's add one more well here, just the DNA by itself. As it electrophoreses, it's small relative to proteins, so it all winds up at the bottom of the gel by the time you're done applying this electric field. If the transcription factor that you've added in these two wells on the left, if the transcription factor protein can actually physically bind to this radioactively labeled probe, what's its molecular weight? What's its size relative to just the DNA by itself? This is where the DNA by itself winds up. We see it radioactive down there at the bottom of the gel. This is bigger or smaller, heavier or lighter.
Heavier.
So protein by itself is...
Oh, that's protein?
Yeah, this protein by itself is big. So if you ran the protein by itself, let me add yet another lane. And we will definitely be talking about control experiments when we look at this. If we just added the transcription factor by itself with no DNA, if we could see where the protein was, it might be that size. It's bigger. It moves more slowly through the gel. So the question is what happens when you combine the transcription factor with these two different DNA molecules. If the protein can bind that fluorescent, radioactive, whatever, labeled DNA molecule, if it binds that enhancer sequence, well, we've added in this lane, we've added the DNA with the enhancer sequence, which is like this lane. So we should still see the same sort of pattern. At the bottom, we should see a bunch of labeled DNA, and that's true in that lane as well. So all of those lanes should look the same because we've added fluorescently or radioactively labeled DNA. They're all going to be really tiny, and they're going to go down to the bottom of the gel. What are we going to see in lane 2 if the transcription factor, the protein we've added, binds the DNA that's in that sample? Here's the size of the protein. Here's the size of the DNA. If it binds, although I should say, we can't actually see, because the protein is not radioactively labeled, we can't actually see it on the gel. We know already that that's about its molecular weight, its size. So if it can bind the DNA, then you should see a band there that is, what that's saying is that's the protein that's bound to this labeled DNA. So the labeled DNA is the part that we can see on the gel. That's why we can observe that band. It's just bigger